Okay, everybody, so we should be in part 2 of our notes about DNA. So we should have just done DNA structure, and you should have learned about nucleotides, and that A or adenosine always pairs with T thymine, and that C cytosine always pairs with G guanine, apples in trees, cars in the garage. And now we're going to talk a little bit about DNA replication. So this should be in your DNA replication notes right under where we ended your DNA structure notes. So again, follow along, fill in the blanks are underlined, and it follows directly with your notes. So first of all, DNA must make an exact copy for 2 new cells. So when we talk about like meiosis and just cells dividing, like when you get a scrape, so like I told you guys I fell on my run, when I get new skin cells to cover this up, then it needs DNA inside these cells because DNA lives in the nucleus of our cells. So when cells divide, they have to have an exact copy of the DNA in the new cell of whatever it's trying to copy or create. Any mistake in the copying process is then called a mutation. So from our videos, this can happen from external factors. It can happen just in our genes. And there are some things that we're going to learn in DNA and little proteins that kind of go through and make sure that there aren't mutations, but they can still happen. And then DNA replication occurs during interphase, which is the growth part of the cell. So that's when the actual copying of the DNA occurs. So this is also called semi-conservative replication. It's just how DNA is replicated. So during the replication, the parent strand is separate and serves as a template. So it's like the original document. Like if you're thinking of a Google Doc, it's like the original template. And then the new DNA molecule is composed of 1 parent strand and 1 new strand. So if we look here, the blue one is the parent strand, and then these purplish ribbons are the new strand. And see how it's semi-conservative because it keeps what the original is, what it starts with, but it also makes a new one. So it's only like half conserved. So it doesn't, you can't keep the whole thing and make a copy. It has to get a new one and have some of the old one. So it's an old strand and a new strand put together is the semi-conservative replication method. So there's just some enzymes that we need to learn about, or some things that happen during the process of replication. And this will go along with a diagram in the next slide. So there's an enzyme called DNA helicase. Any of these things ending in ACE, ASE, is an enzyme which is just a protein, which we're going to learn more about how proteins are made. But just so you know, enzymes help a process go faster. So things like copying the DNA will work faster if you have things like enzymes in it. Same with chemical reactions. When you get into chemistry, it makes the reaction go quicker. So DNA helicase actually helps unzip and unwind the DNA from the double helix. So remember from our gizmo, how we said it copies by unwinding and untwisting. So it can't be, you can't copy something that's all twisted up like this. You want to unfold it and then unzip it so that you can make an identical copy. And then DNA polymerase is another enzyme that catalyzes or creates the correct nitrogen bases. So it's going to pair, you know how A pairs with T, it's going to pair those correctly on the new strand. The leading strand is just the one that's elongated continuously as the DNA unwinds. And then the lagging strand is one you'll see in a video and in the diagram. It is copied or elongated discontinuously. So there's just little parts. And those parts end up being called Okazaki fragments. And that's in the last bullet point here that says joining. So DNA ligase is like the glue, and it links Okazaki fragments. So here is the diagram. So we start with number 1 over here where, excuse me, DNA helicase, the enzyme, remember it's just a big protein. So these purple, like 2 oval purple things on the DNA. It unwinds the parent double helix. So the blue one is the parent. These purple enzymes are going through and unwinding and unzipping the DNA. So it actually started from this side and went in towards the parent strand. So then number 2 is the single strand binding proteins. They're just these little orange guys. You don't need to memorize them, but there's just this thing called a single strand binding protein. It just stabilizes the unwound parent DNA because the chemistry and the bonds are very unstable, so it wants to keep it stable so that it's able to copy. Then number 3, the leading strand. So remember the one that's copied continuously is up top here. You can see the DNA polymerase is just going through and copying everything. So it's called synthesized or made continuously in the 5' to 3' direction. That's just how the DNA is lined up. So if you take IB Bio, you'll need to know that. But it is synthesized or created by DNA polymerase. Then number 4, the lagging strand, the one down here. Remember how it can't be all copied continuously? So the lagging strand is synthesized discontinuously, so in parts, where primase, DNA primase, one second. There we go. So DNA primase synthesizes a short RNA primer, which is extended by the DNA polymerase to form the Okazaki fragments. So you can think of the DNA primase as these little squares. Or sorry, the squares are the DNA polymerase. I don't know where primase is here, this big block right here. So it just starts or shows where the DNA polymerase can come through and put on the matching base pairs. And then number 5, after the RNA primer, which you don't really need to know what it is, but it's kind of like a flag that signals where it can go. So after the RNA primer is replaced by DNA. DNA ligase then is, remember that's the glue, comes and joins the Okazaki fragment. So here's DNA ligase, the circle, it's joining these two fragments. Because if you see, only this like pink area can be replicated to the blue area, and then this pink area goes down to the blue area. Versus this top here, that can just go in a straight line. So the backwards side or the other side, the lagging strand, has to be broken down into parts. And you'll see that in this video that I have for you. And again, the overall direction of replication is going this way or through the opening. So the replication fork shows where helicase is unzipping and unwinding the parent strand of DNA. So then we have our video.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called Primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
All of the names of the proteins or the enzymes in that video, just be able to understand how it gets replicated. So you don't need to know about like some of those RNA things that she said, and some of the other ones I didn't even learn till college. So don't worry about that. Just take in the visual of how the Okazaki fragments got joined together, how DNA polymerase unwound the strand of DNA. Just stuff like that, and you'll be good to go.